Am I one of those people? No, okay, Quinn.
No, Natalie and Quinn are recording. It's okay,
I've
Marsha.
started recording.
Okay.
Alrighty.
I have also started recording.
Okay.
Nice.
Awesome.
Hello, everybody, and welcome to our 5th webinar. We are the Saskatoon Youth Climate Committee. My name is Elizabeth.
I'm Natalie.
Every time. I'm Quinn.
Do we want to just?
Do you want to just redo it? Okay, Marsha.
All right.
That's a good idea.
All right.
Okay, we'll just do that same order, and then it'll be Marsha, and then it'll be Elif.
Awesome.
Everyone good? Okay.
Natalie, you go before me.
Okay. Hello, everyone, and welcome to our 5th webinar. We are the Saskatoon Youth Climate Committee. My name is Elizabeth.
I'm Natalie.
I'm Quinn.
I'm Marsha.
I'm Elif. This week, we will be discussing invasive aquatic species with our special guest, Ian Phillips from Water Security Agency. Ian, can you tell us a little bit more about yourself and what you do?
Thanks, Elizabeth. I'm the Senior Aquatic Macroinvertebrate Ecologist for the Water Security Agency of Saskatchewan. I've held this position for about 13 years now, and over the course of doing so, I completed my PhD, and I'm now an adjunct professor at the University of Saskatchewan, as well as a postdoctoral fellow with the Institute of Global Biodiversity in Berlin, Germany, where I came back this spring before the COVID hit. My role with the province, it may seem by those intros as academic, but it's an intersection between an education role and that of management. So I have a mandate from the province to manage, or at least better understand, aquatic ecosystem health in rivers, streams, and lakes, and provide solutions to improve that health, monitor impacts from activities, and then try to find ways to remediate it to a healthier ecosystem. The reason I have my job is that the prairie ecoregions are less studied than others in Canada for their biodiversity in aquatic systems. And the tools to monitor ecosystem health haven't been well developed yet. And as we increasingly use the resources we have on the prairies and are looking forward to climate change on the horizon, a better understanding of those ecosystem health measures is needed in order to make sure that we have good quality surface waters for the future. So what I was intending on presenting today is kind of a background of what I have created, the importance of aquatic invasive species, and where I want to go with my work in the future as it relates to climate, as best I understand the mandate of the Youth Climate Committee. Does that help, Elizabeth?
Yeah, that's awesome. I think that we lost Elizabeth, so we'll hope that she comes back soon. Are you able to share your presentation?
I sent it to Elizabeth. I'm on my phone and I have my presentation on a computer in front of me, but if somebody else is able to.
Yeah, absolutely. I can start that for you if you're ready to go.
Yep.
Awesome.
So then is it okay if I say next slide?
Yep.
As we go along?
Awesome.
Super. Well, when you're ready, let me know.
Awesome. All right, you can go ahead.
All right. So I titled this essentially Forecasting Changes in Aquatic Ecosystem Health in the Water-Stressed Northern Great Plains. And that's where we'll get to, but I'll start with, in this presentation, how we assess ecosystem health and really what that means to begin with, and then get to where we want to go for how we can forecast what the impacts of climate change will be. Next slide. So the objectives for ecosystem health are to construct a biomonitoring program that is capable of detecting human impacts. Typically, when we think of biomonitoring programs, we simply want to know, is the ecosystem okay? Is it healthy? Has a human activity damaged it? However, there's some fundamental foundations that need to be built before we can get there. We need to know what parts of the ecosystem are actually sensitive to human activities and in what ways. So the key word in that first paragraph is forensically, and that's the gold ring or gold standard of what we want to achieve, where we have measures of the aquatic ecosystem that are forensic to different stressors. So we'll have, as a goal, some bugs that are sensitive to oil pollution, whereas others are sensitive to organic inputs from wastewater, for example. And when we collect the organisms in a water body or observe them, we'll be able to tell where the sources of stress are coming from and be able to mitigate that. As a course tool, if it only tells you that it's impacted, there might be a lot of activities on the landscape and you don't know which one is actually causing the impairment. For example, in the Wascana Creek near Regina, there's nearly 80% crop agriculture in the upstream watershed. There's the city of Regina itself with urban runoff. There's wastewater coming from the water treatment plant and hundreds and hundreds of road crossings upstream as well. So those are just a few of the stressors. But if the site is impaired, we want to be able to forensically identify where that impairment's coming from so we can focus our management efforts on that. The big part of that is really surprising to a lot of folks, and it's simply to establish what we have in Saskatchewan. So for our biodiversity, the aquatic macroinvertebrates, or that's the bugs that are in the water, are really quite understudied. There's been some fantastic work done in the past, but they're a highly diverse group of organisms. And it wouldn't surprise me if through the course of this work, we discover new insects to science. We already have many that we've collected that have no names. And so the next step with that is to give them names scientifically. But that's a secondary academic role that I'll have students help me with in the future. As it stands though, in order to understand what the human impacts are, we kind of need to know who we have first before we start looking at it that way. Further, aquatic invasive species can really disrupt these ecosystems. There's a difference between a range extension when an organism moves into Saskatchewan because it's expanding its range due to post-glacial recolonization, or a change in climate. And when something's invasive though, it really impacts the ecosystem or economic reality of the people using it in a negative way. And that's where we really try to characterize the term invasive. So some of the more common examples people be familiar with are zebra mussels or rusty crayfish, organisms such as that. And I'll talk about that later on. And finally, where I really want to get to, and where I went this winter to work in Berlin with the Leibniz Institute of Global Biodiversity, was on how to use modeled changes in climate that we have for the prairies and other parts of the world to predict the global warming consequences on biodiversity. So if we're able to understand or at least predict what the changes will be in the future due to climate change and the amount of water we have and where it'll be, I should be able, given the knowledge that we have developed, make some predictions on what will be the impact on the amount of insects we have and the forage resources available for fish and birds. What our expectations for biodiversity in the prairies would look like. Next slide. So the foundations. Really, if there's any takeaway to begin with that you should have to understand how we build a program like this, it's what's the reference condition we want to expect in an ecosystem. So if we had a second planet, Earth, theoretically, that had no human activity on it, we might be able to understand by sampling those streams what type of insects we would expect in Saskatchewan in the absence of human activity. But we really don't have that, and particularly on the prairies, there are very few watersheds and rivers that do not have human activity. There's a couple like Rock Creek down in the Grasslands National Park that are really without much human activity at all. But setting that reference condition requires a lot of replication, a lot of different sites that have different characteristics and different macroinvertebrate communities, so we understand the range of variability in what we want to expect. Really, the invertebrates that live in Rock Creek are quite different from those that live in the South Saskatchewan River here and those that live in the Wascana Creek near Regina. And so we need to establish that baseline to begin with. Further, that expectation for reference condition really relies on a philosophical sort of paradigm. if we set our expectations to be before Europeans arrived and what the streams looked like, and before we started that crop agriculture, that might be ideal, but we really don't have any invertebrate collections made from that time period. People were either busy buffalo hunting or as things transitioned into European colonization, into agriculture, and there really wasn't much of a historical record dedicated to the aquatic macroinvertebrate fauna of the prairies. So we don't know what that looked like. But as it is also, there was a human activity at the time too. First Nations burned quite a bit. There was bison herds that traveled through our water bodies and used that limited water resource on a dry landscape. So really that's characterizing the bug community that would exist given those activities at the time. And that leads us to today with what we could possibly achieve for understanding what we should have. And what we're using in Saskatchewan is the best possible reference condition. So I'll walk you through how we establish that. But essentially, what are the best sites we have with the least amount of human activity, and what the macroinvertebrate communities look like there. And once we know that, then we can take a site that might have oil pollution or organic waste from a wastewater treatment plant, and then understand how it's impacted by comparing it apples to apples, essentially, to sites that don't have as much human activity, and what could we achieve at that site that's impacted. It also provides the opportunity where sites, through rehabilitation or reclamation, can be even better than the best possible we have, and they can have better ecosystem health than those that do have some amount of agriculture in them. So in other parts of North America, in the States, for example, they have the Clean Water Act, where they've developed legislated guidelines for what ecosystem health should be in their water bodies. The European Union has the Water Directive Framework as well. And in those systems, a lot of work was initially done to focus on watersheds that really don't have human activity and do have quite a high diversity of different orders of benthic macroinvertebrates. Here in Saskatchewan, though, we don't have legislated targets, and the reason this sort of tool hasn't been developed to date here in Saskatchewan is that there is this human activity. In Canada, the first efforts for it were in the Fraser River Valley, for example, where in some of the higher up watersheds, there wasn't any human activity there to speak of necessarily, so they could set their expectations to least amount or zero human activity and what the macroinvertebrate community should look like. So we have a challenge here in Saskatchewan, but I'll show you how it's how we overcome it and how we have a tool that we can use going forward. Next slide. So as I mentioned, reference condition, it's not your dad's mountain stream. When these original tools were developed in other parts of the world, they were able to access streams that had very little human activity. So on the left picture there, you'll see a stonefly, and that's an insect that's very sensitive to low amounts of dissolved oxygen. It's very sensitive to habitat disturbance or increases in temperature. And it's an ideal candidate for, if we do have a human activity that's negative on the ecosystem health, that insect will respond quite quickly. On the prairies, though, we don't have many stoneflies. There are some exceptions like the South Saskatchewan River that brings water from the mountains. But for most of the rivers and streams across southern Saskatchewan, we don't have these groups of organisms that are typically held by the previous generations that developed these tools as being the unicorns of a water body, the really sensitive and unique organisms. Ours are a lot more hardy, a lot more robust, and the diversity has evolved here with a lot of more extreme conditions. So in the right-hand picture, I have a caddisfly, which in other systems is held to be sensitive to human pollution as well. But the species of caddisflies we have on the prairies here are pretty robust. They've evolved to adapt to temporally changing hydrology, where it might dry out in a water body, it might flood, the temperature extremes are quite high. And I really like this picture as it illustrates that caddisfly is such an enterprising little beast. He cut off some of the flagging tape from one of my experiments and adapted it into his home that he lives in. So that really, to me, sort of characterizes the type of macroinvertebrates we have. They're typically hardier, and they're sensitive to stressors and pollutants that those stoneflies and the previous generation of biomonitoring would have considered a whole different scale. Next slide. So this is what we often deal with, long expanses of prairie where there's very little forested vegetation. And the forested vegetation we do have is often right along the water bodies if it exists there. Next slide. And an ideal candidate stream for biomonitoring is something like this. That's a former student of mine, Alex, who's sampling macroinvertebrates in a riffle. And that would be ideal. That's the type of habitat that these tools were developed in elsewhere in the world. Next slide. But typically in Saskatchewan, we have water bodies that dry periodically, or they have very little treed vegetation surrounding them. So the inputs of what we call alochthonous material, or leaves and sticks that would feed the macroinvertebrates, and the shade that would be provided by the trees to keep the river cool, it just doesn't exist. And naturally it wouldn't have existed. We think prior to human or European colonization, there's very few trees on these water bodies anyway. And so the macroinvertebrates had to evolve in this landscape. Next slide. And now that European colonization has arrived, in this slide, that's a colleague of mine, John Mark, who's walking down the length of a stream that's underneath that grass. And the crop agriculture has gone right to the riparian edges of the stream, really. That's what would have been the beginning of the stream historically up near those spruce trees in the background. And it's left of a former homestead. So not only did they have extremes to deal with naturally, but on top of that, we have modified the landscape, drained wetlands, and added crop agriculture that really adds nutrients and takes away some of the natural processes right to the edge of the streams often. And this might be, next slide, here's a Qu'Appelle River in its upper reaches. This might be the best we expect for a river in Saskatchewan. It has some sound riparian vegetation and willows and other grasses and sedges, but it doesn't have an overhanging canopy of trees. It gets a lot of exposure in the summer and in the winter. Next slide. And in the winter, it's a whole other extreme for these organisms. Sometimes the rivers and streams freeze right to the bottom in a lot of cases. And the organisms needed to evolve ways to tolerate that. We have students working with us who are focused on how these organisms overwinter and how they've been able to evolve to optimize this whole situation and migrate to new habitats when they open up in the spring. It's some pretty fascinating work. You should have them come talk about in the future. Next slide. So when we start developing a biomonitoring program, this is really what I have to work with in Saskatchewan. That if we picture that red dot being the site that we're interested in the ecosystem health of, if you follow that river network in blue, really the landscape is a patchwork of crop agriculture and road crossings and modification. And so trying to find sites with least amount of human activity was quite a challenge. That was the initial beginnings of this. Next slide. So that's the background. Now I'll get into how we actually achieve this and how we apply it. And brief, sorry, I'm just going to decline a call. Is everybody still there?
Yep.
Cool, I see a nodding head. All right, Bont, we'll start with how we assess ecosystem health on our big rivers and work down to our wadable rivers, our smaller systems. So this is Gardner Dam, north or south of Saskatoon, upstream on the South Saskatchewan River, and it's really had quite an impact on the river. It has some very useful human applications. We generate a lot of hydroelectricity from it. There's irrigation that comes off the water body as well as recreation. Its impacts, though, are myriad, and what we're trying to do at the Water Security Agency is better understand those specific impacts to the river and how we can mitigate them, as well as better manage the reservoir for the future. So my purpose in all this was to find out, can we use the macroinvertebrates to understand how the dam and its impacts affect ecosystem health, and what sort of recommendations we can develop to mitigate that impact. Next slide. So for a bit more context, with this large river biomonitoring, these are the two main large rivers in southern Saskatchewan, the North Saskatchewan River and the South Saskatchewan River. They come together near Hey guys, do you mind if we pause for a second? I hear my son calling me.
Oh yeah, that's okay.
I'll be right back. Yeah, Ragnar?
We'll just cut the video here for
Yeah.
this editing. Yeah.
Yeah.
Has Lizzie popped back yet?
Yeah,
I
I'm
think-
here.
Okay.
My phone just like totally glitched out, and I was like, ugh.
I know, I saw that and I was just like, okay, I'm gonna pull the questions out. I'm ready to do this.
I was just super stressed that like it was going to like stop because I was host, right? I was like, oh no, everyone's just
Oh,
been kicked
yeah.
off of the meeting. That was my worry.
Yeah.
Problem
It's
solved.
to someone else. That's nice.
Are you guys ready to continue?
Oh, definitely.
Yep.
All right. So with the large river biomonitoring, the two main rivers come together east of Prince Albert at the forks of the. I gotta go another issue.
Kids.
Oh boy.
Maybe
Yeah,
we'll
just an
rest.
FYI, his son is like 4, so
Oh,
that's why it's like
Okay.
critical. Yeah.
Yeah.
I just thought I would explain that.
Yeah.
It's not like he's like way
makes
older,
sense,
but he's
yeah.
like 4.
Yeah, no, that's totally cool. I understand.
Yeah.
Woohoo. How many sides?
I keep bumping this banjo beside me, and it's not a good time.
A banjo?
Yeah, see?
Oh my god.
It's a banjo.
You are so cute. Oh
Wow.
my god.
I'm just stressed you can like hear these like strings being like hit.
Oh.
Sorry, everyone. An issue inside with my son.
It's all good.
Okay. Trying to childcare and do science is challenging in these modern times. Well, and that's the context. To understand the Gardner Dam and the impacts that it has, we really don't have more large rivers in Saskatchewan coming from the mountains that we can use to set as an expectation what the river should look like. So with that said, the focus of this work was to use other sites in the Saskatchewan River system that were unaffected by Gardner Dam on the North Saskatchewan River and upstream of Gardner Dam to understand what the natural system would look like in the absence of the dam, and then provide the recommendations off that. Next slide. The biggest impact of the Gardner Dam, in addition to hydro peaking and some of those other more subtle dam typical impacts, is actually temperature, believe it or not. But the way that large reservoir in the areas like Gardner Dam works is that in all these big lakes, you have a warm surface temperature we call the epilimnion, and a cold bottom temperature of the lake called the hypolimnion. And so each summer, the top of the water warms and the very bottom of the lake stays quite cool. You'd notice this if you're swimming in the lake and the surface is pretty warm, but you put your feet down and you notice that it's quite cold down there. Well, the way the dam works is that the water comes out of the bottom of the reservoir through some turbines and uses the pressure on top of it to create that potential and the electricity that we use. However, that water coming out is quite cold. And in these figures here, there's temperature on the y-axis and time of year on the x-axis. You'll see I highlighted a couple reference sites and put Gardner Dam on here to illustrate how the temperature is depressed. We did this by having a couple rather entertaining summers deploying temperature loggers all through the Saskatchewan River system and really living out of a boat, collecting macroinvertebrates and measuring temperature. It was quite a bit of fun. But the orange line there, dotted line, is the temperature of the South Saskatchewan River at Leader, upstream of the Gardner Dam, whereas the thick rouge line, I think it is Borden. And those are two sites that are really unaffected by Gardner Dam. It's either upstream or it's in the neighboring river in the North Saskatchewan River. And they have a typical normal sort of distribution of temperature over the course of the summer. It's warming from late April up to a peak around July, and it starts to decrease as we head into fall. However, in the downstream of the Gardner Dam, there's that light green line that heads up with inverted triangles, and it's slower to accumulate temperature over the summer as the reservoir takes a long time to warm, and it really only reaches a maximum around 15 degrees Celsius in late August. So that dotted line with the title there, Hexagenia limbata, illustrates if you're an organism living downstream of the dam, you'll need a certain amount of energy in order to grow and reproduce and emerge from the water body. However, if it stays cool through the spring and doesn't reach the same peak, there's not as much area under the curve or as much thermal energy to allow you to complete your life cycle. So that's the same for, on the right panel, Orconectes virilis there. And it's really that cold environment, it's refrigerating the macroinvertebrates, and you end up with a different group of invertebrates that are able to tolerate cold temperature, very different from the invertebrates that would be there in the absence of the dam. Since I was asked to comment on the theme of aquatic invasive species, this adds an element of risk too. On the right panel, B there, Orconectes virilis is our native crayfish, and it requires a certain amount of energy for its eggs to hatch at around 11 degrees Celsius. Whereas the invasive crayfish that hasn't arrived yet, but if it were, its eggs are able to hatch at about 4 degrees Celsius. And so if it does arrive, there's quite an element of risk where the ecosystem doesn't have other crayfish to compete with it, and it has an open, wide environment frontier that it can really take off in. And that's one of the hazards that we're looking at for the future with that kind of environment and impact. Next slide. So conclusions, a lot of the sensitive organisms that we have from the Saskatchewan River system are gone downstream of the dam, but a group of chironomids, or Diptera, actually took off. And for folks who aren't that familiar with aquatic macroinvertebrates, this group, this family of aquatic insects is we call it a freshwater midge commonly. If you've ever been riding your bike in the summertime through a park and you get a cloud of little flies in your eyes and up your nose, that's this organism. They grow fast, they're very small, and they emerge en masse for a very short period of time where they reproduce and then return to the water bodies. They're crucial in our rivers and streams, actually. They form really the base of our food webs, and they feed our fish. And when they emerge, they really are effective at supporting our songbirds and other organisms like that, bats, that need stuff to eat. So this test site analysis, we call it, and reference condition approach, worked well to be able to identify the impact of the dam. And we've been able to use it since for other stresses, such as oil pollution. If you think back to the Husky 16 pan oil spill in the North Saskatchewan River in 2016. Oh, good Lord. Sorry, I'll try and find another spot to talk. All right, is that a bit better, everybody?
That's awesome.
All right. Can you see me?
Mm-hmm.
So one of these chironomids taking over downstream of the dam is that they're chironomids we did not expect. They're what we call stenothermic midges, and they originate from high alpine or Arctic environments. They weren't known from Saskatchewan before this, but somehow they colonized all the way from the Rocky Mountains or Northwest to this environment, and they've taken over downstream of the dam. So one of the unique characteristics the populations there and support fish in their own way. Next slide. And next slide for biomonitoring for the Northern Great Plains. We come to our application for smaller rivers and streams. We've covered the Eagle Creek, Swift and Saskatoon, the Power and South, that sort of thing. Next slide. 4 main steps to these biomonitoring approaches. The first is to characterize the biological and physical chemical attributes of minimally impacted. As I mentioned at the beginning, you collect those sites that have the least amount of human activity, and you group those benthic macroinvertebrate communities together and try to identify what physical features distinguish those groups. So the easiest analogy I have found to describe this for most people is if you think of ecoregions terrestrially. So you have mixed grass prairie, you have aspen parkland, and shield boreal in the north, for example. In a plant environment, you wouldn't expect spruce trees to be growing on a mixed grass prairie, and likewise you wouldn't expect a lot of the grass species you have on the mixed grass prairie to be growing in the shield, or at least dominating. So really what defines those is the superficial soils and the precipitation in Saskatchewan. When it comes to aquatic macroinvertebrates, it's the same idea. We need to understand what physical characteristics define the bugs that are going to be there, and what are the bug ecoregions, put it that way. So once we have those ecoregions, when we have a site that we think is stressed or may be impacted by human activity, we use those physical features to match the most appropriate test sites to that group of reference sites. Back to the analogy, we would use perhaps the precipitation and geographical region that a site's in terrestrially to identify what ecoregion you'd expect that terrestrial environment to best look like. Then finally, we compare the macroinvertebrate community to the reference sites to see if it's a good match biologically. If not, it may be stressed or impaired by human activities. Next slide. So this involved collecting macroinvertebrates from sites all over Saskatchewan. This is presented a subset of them on this map here, but really it was quite a bit of work over a few years. Next slide. And once we collected the macroinvertebrates from each site, we had to delineate the watershed upstream of the site and then quantify the superficial geology of the site, as well as the land use, the amount of road crossings, all those other physical characteristics that are upstream in the contributing watershed that would create the environment that the bug has to live in. Next slide. And unfortunately, or realistically, the best reference criteria we could come up with in Saskatchewan was a site had to have no less than 5% urban land use, less than 40% cropland, less than 50% pasture, less than 70% total land under human influence, and no greater than 2 landfills, oil wells, bridges, or road crossings. So that is a lot of human activity, really, but that's the best we could find. And it speaks to how well developed Saskatchewan has become. We arrived at a final grouping of 88 reference sites that we were able to work with. Next slide. And once we get to sites that are impacted, this is an actual site here in Moose Jaw that has a lot of oil pollution from a local refinery in it. We're able to take the macroinvertebrates and compare them to those respective reference sites that it should look like. It's worked well, and as a conclusion, sorry, next slide. The Northern Great Plains have this unique paradigm of community structuring and abiotic characteristics. Turbidity makes quite a bit of difference and influence on the expression of the macroinvertebrate community. As well, the reference condition is possible to apply here and successfully detect anthropogenic perturbation. And thus, we called this system the Saskatchewan Condition Assessment of Lotic Ecosystems. And there's much more detail at a technical level into how this works, but for these purposes of introducing everybody to the concept, I thought I'd try and keep it as general as possible. If you're eager to follow up with me in the future, I'd love to hear it. So going on to the next big theme, next slide. It's in the attention of this biodiversity and what we have. There's that handsome little caddis flag again. Next slide. Often large changes in our ecosystems or undercutting of biodiversity goes unnoticed until a tipping point is reached. We may not know what we have as macroinvertebrates in rivers and streams in Saskatchewan, And unless we look, it's really not a problem. It's like the ostrich that keeps its head in the sand. But then all of a sudden there can be an ecosystem change. And we've had this periodically in Saskatchewan in the past with insect outbreaks, with loss of services. After the Gardner Dam was initially built, there was huge, I don't know what to call them, really almost biblical swarms of black flies that took off in that new environment and caused quite a significant amount of livestock damage for a number of years. And it came out of nowhere for people at the time. So this biodiversity knowledge is crucial in order to understand what we have and foresee some of the problems we might come up with. We need these organisms to clean our water, filter out bacteria and other pollutants, and process those pollutants, and they feed all the other organisms that we use economically, like fish and birds, as well as pollinate our plants, that sort of thing. So knowledge is really crucial, and then taking action on how to improve the biodiversity and protect what we do have. Next slide. These are a few of my favorites I thought I'd put up. I think you have a picture of somebody with a fish right now.
Yeah. Yep.
I skipped a few. Okay. Well, let's leave this one out. There's a couple that should have come up before it to give it more context, and now it's just somebody holding a fish.
You missed one, Natalie.
Next slide. Go forward one more.
There we go.
Yeah. So what's involved in this knowledge is really having young scientists engaged with collecting macroinvertebrates, either academically or working for the government, but building up that knowledge of biodiversity. Next slide. And for action, this is an example of where one change I'd like to see in my career here in Saskatoon. This is, I worked with a group of other gentlemen in Saskatchewan a few years ago to come up with a new design for the weir. As it stands, fish can't pass the weir. It impacts the aquatic macroinvertebrates as well. The mussel diversity is highly restrained upstream of the weir, as they can't get through either, and the fish that would otherwise carry them, the squawkidia, can't get through. So we've been pushing politically for this engineered solution where the weir could be maintained if it's desired by other proponents, but then a better functioning fish passage could be built along one of the shores that would allow for that connectivity. As one example, it could double as a whitewater park, and we've been working with the local recreation representatives for that, so that it could have dual purpose. But really, we need to bring the velocity of the water down to about 1 meter per second, so that sturgeon and some of those other larger fish are able to make it through. Next slide. And this, I would love to talk about more with quite a bit of passion. This is the architectural mock-up of what I'm really calling the Great Plains Experimental Rivers Area Research Facility that would be built at Gardner Dam. Now, there's a long history to a rock that was important in First Nations culture. And as I'm not First Nations myself, I really shouldn't belabor its interpretation, only to say that it existed near the Akito Valley, what is underneath the Gardner Dam and reservoir currently, for millennia. But then with the inception of the Gardner Dam and the flooding of the valley there, it was removed from First Nations use, and it was very important in their traditions and their spirituality. It was actually destroyed in the construction, so it can't really be drained and then the rock recovered. It was an enormous glacial erratic, but for the future of engagement, I see building this research facility and targeting both youth and First Nations in education, all the while being able to conduct research on these specific forms of pollution at the tailwater of Gardner Dam going forward. Next slide. And really finally into where we go with this, the with this understanding of the biodiversity we do have comes the understanding of the biodiversity we don't want. There's a lot of species that are starting to arrive because of human activity. Here is a dating app parody that a friend of mine created for Prussian carp on Finder rather than Tinder, and all got a good chuckle out of it. But Prussian carp have since arrived in Saskatchewan. They exist in the Lake Diefenbaker. They're goldfish type carp, and they reproduce prolifically. So it's one we're trying to manage with the Water Security Agency, and it's going to cause us some problems in the future. However, next slide. There's some we don't have yet that we can focus on. Zebra mussel is one example, but here we have marbled crayfish. And again, it's a parody created by my friend James, but on catch.com. We really don't want marbled crayfish in Saskatchewan. It can cause its own problems. Next slide. Or flowering rush. It's starting to move up in the South Saskatchewan River Valley into Saskatchewan, and we really need some engagement and action on the ground to prevent these organisms from arriving and from taking hold here. Next slide. To the future and beyond. So take these foundations, and my goal is next to redevelop the biomonitoring program to really focus on midges, since they are one of the most common groups that we have here. Beyond that is where I really get my most excitement these days. With the great work that's gone on for developing hydrology models in the future of climate change, it provides an opportunity, now that we have a better understanding of the biodiversity of the prairies, to project what the implications will be for aquatic biodiversity. And so that was why I was in Germany, and it's really the next step. So as the climate change projections establish what the stream permanency will be, the amount of water that'll be in them, and their temperatures, we'll be able to set expectations for what the impact will be on the amount of benthic macroinvertebrates we have that support our fish and our birds and process our pollutants. We would like to be able to understand first what it'll be before we can start to take action to see if we can mitigate that. Ideally, we wouldn't have this human-induced climate change in the first place. But again, knowledge is really the first step. Next slide. And this is really what we have at Gardner Dam currently, these freshwater midges that are really cold tolerant. But going forward into the future, these midge communities are the most responsive, and we expect them to change with climate change. And they can be an indicator group for what, when we're reaching tipping points, when we're arriving into new ecosystems, and what we can work toward to achieve by repairing temperature, pollution, and other stressors, and arrive back at a community of midges, or assemblage of midges, that better reflect what we want in Saskatchewan. Next slide. So this is just a thank you to many of the people that have worked with me in building this over the years. I work for the Water Security Agency, but I also am involved in a group called Trout Reach that Elizabeth joined last summer. And it's an NGO that's meant to train young students in this and other scientific endeavors, and work on these foundations going forward, so that there's a better trained cadre of scientists out there who are able to build this knowledge going forward. But really, that's what I prepared today, everyone. If you have any questions for me, I could try and answer them before I go.
Yeah, thank you so much for sharing your knowledge and expertise. I'm sure everyone here learned a little bit, if not a lot. So yeah, if anyone has any questions they want to ask, we have time for probably a couple. Does anyone have anything they want to start with?
I just wanted to ask you about your journey and what really inspired you to go into this, and just a bit more about your background.
Sure. Frankly, really, it was mentors in my life along the way. I've always enjoyed the environment, as I think most young people do. When I was a teenager, I was very active in outdoor activities, but I never saw entomology or ecosystem science as a opportunity for a future, if that makes sense. I really wanted to be a firefighter coming out of high school. But along the way, I had mentors that encouraged me to try university, and then gave me jobs as I started university, and encouraged me to continue on this path. And they were really foundational in allowing me to continue to get into an honors degree and into a master's and into a PhD, and provide me with the best advice and encouragement along the way. And I think if I had any advice for young people who are starting out, taking on these paths alone is admirable, but really there's, I think in any discipline, particularly in this discipline, there's a lot of scientists who are eager to take on enthusiastic young people in one form or another to provide them with mentorship, if that makes any sense.
Thank you.
Oh, sorry, Lizzie, did you have a
No,
question?
go ahead. I was
Okay,
just going to
sorry.
say, ask if anyone else had a question, but you do, so go ahead.
Is there anything that like we could do as individuals to support like indigenous like aquatic biodiversity?
There's probably a ton, and it gets quite overwhelming at times. Like a lot of environmental issues, it can be defeating to think about it as a big picture. Really, after the time I've spent in this field, knowledge is crucial. So educating yourself on what exists helps build a foundation yourself for what's in the environment. But then also understanding the laws and regulations that allow us to use those resources, and have activism that's not just yelling into the sky, but focused on specific aspects of that to improve it. It'll be exhausting to think that we won't have an impact. We will. We're human beings, and we have such an industrialized commercial society in our modern world that there will be effects we have on the environment. But with all those activities, there's regulations that provide for how oil and gas is extracted, for how agriculture is conducted, the guidelines for what level the outflow constituents from our wastewater treatment plants need to be at. And if there was a career that I'd advocate anybody go into in order to have a positive effect on the environment, really it would either be law or politics. Coming into science and working on these issues like I am is very rewarding personally, and it is helpful, but those regulations and laws and engagement with other groups that are underrepresented in our science, such as First Nations, really isn't my job necessarily, nor am I well educated in that area. So there's other NGOs such as Eco Justice, I think it's called. They're pretty effective in holding government to account for their environmental standards, and that sort of engagement I think is pretty important for the next generation coming along, if that makes sense. But on a daily basis, the Entomological Society of Saskatchewan is always eager to have amateurs and young folks join it. There's the Saskatchewan Wildlife Federation, short reach as well, from my own perspective. But there's environmental groups that are eager to support and provide mentorship to young people, whether it's Nature Saskatchewan, the Saskatchewan Environmental Society. There's quite a few, and they'll help people get started in understanding how to identify the nature they have and how to understand it better.
Alrighty, so do we have any more questions? I would say maybe one or two more. Does anyone else have any questions they want to ask? No? Okay, I just had one question about, you talked a little bit about the importance of the riparian zone, and specifically around agricultural lands and crops. I was just wondering if you could give an overview as to what people who have those lands could do in order to mitigate the impact they have on water systems, and then in more of a general sense, what we as individuals can do to prevent our impacts as well as the spread of invasive aquatic species.
Sure, I'll do my best, Elizabeth. Well, to begin with, it's really an easy solution. In a way, on a practical level, what I've found with my research is that the amount of uncultivated land you have in the contributing area upstream of a watershed really explains the ecosystem health you have on a landscape level. So translating that into simpler terms, the more riparian vegetation you have and the wider the riparian strips along those water bodies, the healthier the rivers and streams will be. On a more specific level, we've done lots of experiments with pesticides and macroinvertebrate community response. And what we find is, pesticides such as neonicotinoids do affect the macroinvertebrate health. But secondarily to that, riparian areas are very effective in mitigating that impact. So if you have wetlands and rivers that have very little riparian, the big each other with a broader riparian area to help with the water body. How to achieve riparian?
Sorry, could you just repeat what you just said? Your audio was glitching.
Sure, I'll do my best. Really, in simple terms, maintaining the amount of riparian vegetation we have and increasing it is crucial to improving the ecosystem health. Whether it's pesticide or neonicotinoids in particular that we've worked on, but you have greater vegetation, there's less opportunity for the neonicotinoids to make it to the water body. So that's a good example of how we'll still have crop agriculture, but there's other ways we that don't take away our ability to produce nature. How to achieve is a bit of a challenge. There are some regulations about how close you can cultivate to a river or stream, but there aren't laws that are strict and enforced for broad swaths of the province. So really in the future, activism through the appropriate channels with the landowners is probably the best bet. Providing incentives to maintain those riparian areas, engagement through NGOs like Ducks Unlimited and Nature Conservancy Canada are promising routes, and to work with government to try and encourage bit more stringent regulations for that riparian vegetation you have, particularly in headwater streams and wetlands. Oh, he's sticky. Did that? Oh, we'll have to fix that. He had a sticker on his hand, now it's all gooey. If we can use Elizabeth, I think there's a second part to that question. Did I miss it?
No. Oh, I was just asking about what we can do as individuals.
Yeah, and that's say hello. That's the cord. Individuals, cord. The wall.
Oh.
Oh.
And the rest of sisters, those citizens, two people.
Well, I don't know. We'll have to go look for it. I'm just going to finish up and say goodbye, right? And then we got to go fix that sticky on your hand. So, as individuals, there's not a ton. Certainly, some of the more effective acute activities I've seen have been shoreline cleanups. Those are pretty helpful for the health of the rivers and streams. Having worked in rivers and streams my whole career, there's a ton of garbage along most of them, and I pick it up as I go, but through community participation, that would have an effective improvement on our water bodies. As well, really pushing your politicians and your representatives to have that greater increase in ecosystem health protection would be an effective tool. Regulations really are driven by public input and public needs. So if you're expressed as a need, that's really where it starts.
All right. Okay, well, thank you so much, Ian, for sharing your expertise today. We learned a lot. And it was a very valuable learning experience for all of us, I'm sure, as well. So everyone who's watching, stay tuned for our future webinars. We'll be talking about climate change and climate justice and other environmentalism initiatives in Saskatoon. So thank you for joining us today, and we'll see you in the next one.
Thanks for your patience with me, you guys. Hope you have a good afternoon.
No problem.
Yeah, you too.
Bye.
I did just want to kind of double check with what you were talking about there. So sorry, you were saying the cleaning up with the river. So would that be accurate to say that cleaning up in our streets, like where our gutters are, would also be affecting the river cleanliness, because all of that does kind of end up downstream?
Certainly it does, very much so. That and there's another program I was aware of before. I haven't checked into it for some time, but it's called the Yellowfish Road, I think. And paint yellowfish beside stream stormwater outlets to remind people that pollution, whether it's oil that you have in your car or garbage, does end up in the river. That's helpful. But just getting garbage off the street and out of the ditches is enough to keep them out of the rivers and streams. And plastic pollution is a growing concern globally. It isn't just limited to the ocean. Our rivers and streams are found to be full of microplastics and other pollution like that that does originate on land, of course.
Cool. Okay, so anyone who's recording, you can stop recording now. Ian, I just had one more